Okay, I can see there's a few people still rolling in, but I'd like to welcome everybody to this Microscopy Australia webinar on tiny bright things from a great new company come out of Melbourne University. That we thought this would be a great opportunity for them to explain their new halo microscopy to us and for us to all understand these new innovations that could have application to us, of course. So I won't go on any further, but I'd like to welcome Ray D'Agostine from Tiny Bright Things to get started. So thanks, Ray.
Thank you so much. It's really exciting to be to have the chance to speak, particularly since it's Microscopy Australia. I think the last time I saw a lot of folks from Microscopy Australia was when ANFF and Microscopy Australia had a joint annual showcase, but that was years ago in Sydney. So I'm the Chief Science Officer at Tiny Bright Things. Also in the audience is our CEO, Dr. Gregory Ward. And I'm also at Melbourne University, which is where some of you might know me. But today, I'm really excited to talk about a new microscopy method we've developed. We've actually made a product that you can actually see over on the left. It's a microscope attachment that sits on an upright microscope. It uses visible light, and you're able to image objects well below the diffraction limit in air or water. And it works pretty much as fast as you can take a picture. And so today I want to show you a little bit about how it works and then some applications that we've tried out that I thought might be relevant to this audience, particularly if you think about what Microscopy Australia does. And then my goal is to make sure I leave plenty of time for questions. So if you hear an alarm go off from my mobile phone, that's not a call. That's just a reminder for me to stop talking. So with that, uh, let me go ahead and get started. Now, why another microscopy or why another tool? I don't really have to sell to this audience. And while everyone sees the common scale bar we use to describe how different systems work at different areas, may it be SEM, EM, or fluorescence or other metrology tools, well, we all work with microscopy and images. I think anyone in a core facility knows what they're really doing with researchers isn't providing images, but providing insight. And we do that on whatever tool we can use or that's required for the needs of that particular research problem. And so we have an amazing set of tools now that can give us beautiful pictures. But there's always places where we think we'd like to improve or differentiate or had a different way to look at them. And this often comes up if you think about native conditions or delicate samples that might be beam sensitive, or if you want to look at something with a reasonably high resolution, but in water, not in air or in a vacuum. You can also think of things where label free is still something that's a challenge. Some things don't deal with labels well. And then some dynamic processes, even if they are fluorescently tagged, struggle to keep up with a dynamic process as things might break apart or spew out or get it stuck into something. And then we can think of there can sometimes be different limitations by a surface sensitive technique for complex morphology, or you can see inside it, but you don't get a great idea of the outside. So there's still opportunities where to deliver that insight to users. We think we'd like to have other tools available. And that's really why we started looking at the HaloElement. So what we've made is a microscope attachment. Everyone has that great slide of different sizes, and why wouldn't we be ever any different? And we put the general ranges up, and we also put where we think HaloMicroscopy can fit. And what's different here, of course, is all of those images were taken with the HaloElement. And I'll talk about some of those today, but if you're curious about something I don't get to it, please ask. So this is an attachment. It fits on an upright microscope. It has 15 LED light sources, so it's not laser based.
How much?
I think someone might need to be on mute. There's no moving parts to it, and it can do submicron, and nano detection. It works in water or air. It's edge tracing, and I will explain what that means in a minute, but it also acquires the image in less than a second. It normally takes longer to focus on the sample than it does to acquire the image. And so what I want to break up today is a little bit about how it works, and I'm going to step you through some of the key parts of that without hopefully being too boring, and then we'll get to more of different applications. So it uses evanescent waves, and so we use a waveguide to deliver our light. And our sample will just can sit on top of a round glass slide. And we deliver light from 15 different locations that we sequentially trigger to kind of go around the sample like a clock face. Um, the type of light we're using is evanescent waves. And while you can put plenty of complex samples on there, I just for a moment want to talk about samples that have somewhat simple geometries. And so that's what's highlighted there. Now, in that little inset. Now, you might ask a couple things. Why are there a bunch of red dots on it? And why is it in the dark? And so let's maybe cover a couple things about how ultimately this works using evanescent waves. So we use total internal reflection and create visible light evanescent waves at a solid liquid or solid air interface. Now, this, unlike fluorescence where you're doing excitation, this is just using visible light. And for those of you that have worked with evanescent waves, you'll know that when an object gets in an evanescent wave, it frustrates and scatters light. But because the evanescent wave is near field, the only light you see is the light that's scattered from that object. So it will look like a dark field image. Over in this little box, I have a schematic showing of what a sphere might look like as it frustrates an evanescent wave. And then I actually have a bright field image where I've got the bright field light on as well, but the evanescent wave scattering is that little red dot. Now, and so to make an image, we use 15 different light sources. So we get 15 dots around an object. So you can actually get the outline of the object from kind of doing a dot to dot connection of those spots. Now, a couple things about evanescent waves. They're not diffraction limited. So you can scatter and image objects that are well below the wavelength, the diffraction limit. The smallest thing we've looked at or detected is a 15 nanometer gold particle. The other thing is what those hotspots do. In microscopy, it's very easy to find the location of a bright spot. It is very difficult to find an edge of something, depending on the resolution of the instrument. But in this case, these hotspots are actually generated on the edge of the particle. So when your bright spot's on the edge of a particle, it actually becomes a very good way to find the edges of an object. And so these, what we call them hotspots, they're resonance scattering hotspots. I'll get to a little bit more about why they're resonant in a moment. But these hotspots form around the outline of a particle. And so for simple objects, you really can get the outline of particles quite well. Now, what that looks like is this is the HaloElement operating. We're firing sequential lights, no moving parts. It's just a sequence of lights around a clock. And then we have the animated version. But over on the right, we have what it looks like in the microscope. And so this is a series of some 10 micron or 5 micron polystyrene particles. And you can see each frame is that red light going around, firing as we go around the particle. I've gone in there and used green dots to find the center of those hot spots and the outlines, and I even clustered the particles at the end as well. But we're using just one color LED source, and we're able to go around the image. Now, around the particle. To make an image, though, we use all 15 of those images and stack them together. Now, to make you guys experts in HaloVision, the software that comes with this, where we have the attachment, we use a camera and there's a controller, the software does something a little different, and I'll show you, and that's, well, we use just red light. We could use white, but we just use a single wavelength and go around that in a circle. We actually give each color a faux color because of it's based on its position. So there's a false color for each channel because it's located in a different position with respect to the sample. And so then we take those colored images together and smack them together, which is why many Halo microscopy images will have this kind of cool rainbow hue to them, which is actually called phase in our color scheme. Now we do that because there's actually angular information that's encoded because of the locations of these lights. Now I say that, but I'll have an awesome example a little later to show how that might work. But before we do that, now I kind of want to just start to ease into looking at interesting objects. And the first part I want to talk about is, I said this was an edge detection method. And so what we have here is a pattern, of course, in the company logo. I do have to admit there's a Melbourne Uni logo kicking around somewhere as well, where we have the halo image above and the non-halo image, just a simple bright field image below. Now, this was made with EBL, and it's a pattern with a 50 nanometer flat gold film. And it might be a little hard to see on the webinar screen, but the insides of those letters are quite dark, even compared to the background. And that's because halo is not really scattering within that flat isotropic film. It's scattering on the edges because that's that sharp geometry change and dramatic change in refractive index from air to gold. So it's really great at picking up the edges, whereas bright field, you actually are seeing the flat film as well because it's occluding the light. But it's also, since it's good at edges, it can also detect cracks. So we made those samples quite a long time ago. And when we have a couple we've looked after, we had one we didn't look after. And if you let that gold sample sit there for 2 years and not a great environment, it might start to delaminate and form cracks. So this is one of those patterned samples 2 years after a little bit of lab abuse and neglect, and you can see it's actually got cracks in it now, particularly around the G and the H, and the little letters are starting to crackle and come off as well. And so now we're actually picking up the cracks that are in the film as well. Now, I'm not saying everyone should make a great EBL sample and let it sit for 2 years to start to crack, but this just happened to be the example that we found. If we think about other samples, and the example I have, I think, is familiar to most people in plants, is the cross-section of a lily, which is a very common standard. And here you can see, compared to the bright field image, we really are good at picking up the edges of that sample. Now, that's edge detection. But remember, I did say it works, it can collect cracks. And so this is actually a large MOF particle, but it has both crystalline and amorphous regions. The dark triangular region in the middle is probably the crystalline part, but you can see kind of a rainbow color where we're illuminating little lines and cracks in that sample as well. And then the outer is really showing up. And that's really because after talking to this researcher who made the sample, that has both amorphous and crystalline regions. And what we're doing is it's the same material, but Halo's very sensitive to both differences in refractive index and geometry. So that's actually what you're scattering off of. And you don't need a large difference in refractive index to actually start to see contrast and scattering. And geometry, of course, helps as well. And so here we can look at fault detection. But one thing to remember is looking at a crack or a fault, if you have enough cracks or faults, you could have internal structure. And so this is silicon carbide grains. And on the left, I admit that is not a great bright field image of those silicon carbide grains, but we don't have the condenser on when we have the halo in. And then there's a halo image of the same image. And there it looks better because we're getting really sharp contrast on the edges. But also, this is a zoom in of some of those particles where you're actually starting to see the internal crystal grains and structure in that particle. And that's different. You wouldn't necessarily see that in an SEM or AFM because that's a surface technique. I suppose you could do TM, but I think these are too big for that. But you can actually start to see internal structure as well. And again, it can scatter off occlusions and differences in geometry. And so you go, well, how does that work and why does that work? And I'm going to share with you, we did Lorentz-Mees scattering, spent probably years in ComSol looking at this to convince ourselves about edge detection and did a lot of laser systems to calibrate it. Instead of boring you with electrical scattering field vectors, I did want to show you some simplistic ray optics representation of what's going on when you're doing this type of evanescent wave scattering. So we have the schematic up there. Again, this is what a hotspot would look like for a sphere. But down on the bottom, I have a very simple ray optics calculation, but I want you to take away two things from it. First, it might be hard to see, but there's a little line down on the right, or in this three-dimensional image, you can see a plane of the evanescent wave and then the particle. And the take-homes are, the particle doesn't have to frustrate that much of the evanescent wave to illuminate the entire particle. So the evanescent wave penetrates maybe half a micron into the solution, a micron at best, but you can look at objects that are 10 microns tall or larger. And that's because the light resonates, or you could say bounces around, we'll say resonates within that object. And so that's actually how, for an isotropic object, it just bounces around. It doesn't scatter off anything. But imagine for the second if you had an occlusion. Now that could be a crack, it could be a different material, it could be an air bubble. By the way, air bubbles scatter like disco balls. But what happens then is if you look closer in, these are all the same wavelengths. The colors are just to illustrate the origin. But here you can see that the occlusion itself will also scatter as well. And from that internal resonance, even if it's nowhere near the evanescent wave. And so that's kind of why we can image both the edges and internal structure of objects quite effectively. And so with that in mind, what I want to take you through are a couple examples. And because I know there was a lot of interest in EM in Microscopy Australia, I've been a little presumptive to say, hey, maybe in a workflow with EM, these might be ways to look at these samples and think about how Halo could fit into a workflow or add value to an existing one. And the first things we're going to talk about are fibers and flakes, because, oh my gosh, we've really found this works quite well with a variety of fibers. This is an example from a group that they typically make an electrospun polymer fiber for tissue cell culture. They do it with SEM. They characterize it there, and that's what they get the image from. Now, if you wanted chemical composition, SEM would be great for EDX, but are there ways you could get an image or an idea of its geometry without having to put it in a vacuum and possibly code it? And so not exactly the same sample, but from that research group, we also did electrospun fibers with them that similar, maybe not exactly the same electrofiber, but now that's a halo image. And here you can see a couple things. You still see that really big depth of field. Remember though, this is only a 2D image technique. This isn't 3D. But because the light couples into those fibers and is such a great light pipe, you see quite a big depth of field. And you might notice some of the sample looks a little out of focus. That's not because we were bad at focusing the microscope as much as it was a thick sample and we're really illuminating almost the entire focal block. Now, I would never say don't do SEM, don't do TEM, get that beautiful, gorgeous picture. But if you want a fast answer first or find out is that sample worth putting through that workflow, this might be a useful thing to do beforehand. Now, this is another example of one that's quite is difficult to see, not impossible, on a bright field image. And these are collagen fibers in water. Now, this isn't an involved sample prep. We threw the collagen on a slide, put a cover slip on it in water, and just looked at it. This is again used as a tissue culture scaffold. And normally the researcher would actually need to put this on, have a sample that they'd code or put in an SEM to see if they had reasonable coverage. Now, that's not a great bright field image, and absolutely you could do better with a better condenser and a high-res camera. But this is the same optical conditions that we took both of these images at. And the collagen fibers, you're talking 200 to 500 nanometers for those single strands. And because you get such a great light piping effect, you're really quite good at resolving fibers. Now, that was looking at fibers. The other one is not, it's an interesting point about 2D materials. So this is a graphene oxide flake. Over on the left, it's in air, and over on the right, it's in water. Now, if you did the bright field image, you'd still see this is a big graphene flake. You'd see some of it. You wouldn't see all the fine cracks and details that the evanescent waves are really scattering off of as well. But this researcher, Rika Tabor, works with these things in water. And we thought, well, all right, normally you look at 2D materials dried out. What happens when you look at them wet? Graphene oxide has a very complex surface chemistry, so when you put it in water, that surface chemistry causes that flat plate to undulate. And you see a very different structure, and it's quite hard to differentiate because it's a small material, it doesn't have a great contrast for bright field. We're just adding water, you can see something entirely different for a 2D material. So you could imagine characterizing in water and then parallel characterization with SEM might really bring a powerful picture to some 2D materials. Okay, now, what about really crazy samples, opaque ones? We're talking about a microscopy technique. So far, I've talked about using glass waveguides, putting samples on glass slides, but what if it's an opaque sample? And so we set this up. So while we use our round glass slide, not everyone keeps round glass slides around. So we actually set this up so you could use any index matching fluid or a glass one preferably, and actually just index match a glass slide onto that circular waveguide. The whole thing becomes a waveguide, and you can put your sample on there and you can do it in water and put a cover slip on. We've even got Petri dishes that will mate into our round glass slide. So that makes it easy because microscope slides can be cleaned, disposed of, and you can get lots of different samples on them. And that's because we can couple the light into the microscope slide. Well, when you're talking about waveguides, there are opaque things that work as waveguides, namely semiconductors. Silicon wafers are their own waveguides. And then if anybody works here with silicon on insulator waveguides, they'll of course know that. And so what we have as an example is, this was motivated because somebody asked us about a silicon sample, but I'll show you that one at the end. This was motivated by, well, we're not sure if we can do that. So we got silicon samples with gold patterns on them. And where do you find those in Australia? But from the University of Queensland's CMM Center where Elliot Cheng makes these awesome EM calibration gratings. And they're, I think, 200 nanometer high gold, and the smallest line width on them, which I think is up in the left-hand corner, is 200 nanometers. And they're used for EM calibration, but we're just illuminating this with Halo. And some giveaways is we're seeing the edges of the object, not always the solid internal part, because this is a gorgeous made EBL film, or made with EBL, and then I assume sputter or evaporated, really high quality surfaces where you can start to see this, but this is an opaque sample. And what we're doing is we're generating that evanescent wave at that solid air interface. But by the way, you can also do these in water. They actually look a little cleaner or a little less background noise, but you can do these underwater as well. Now, we wanted to compare this. And so this is a field of view calibration where those little spots are 500 nanometers on, I think, a 10 micron pitch, where, again, with the Halo image, we get the outlines of the little squares. But we compared that sample, we threw it in an SEM for comparison. And just in case you haven't found the SEM image, it's actually that part of the pattern where, again, with the SEM, you can see the top part of the gold where we see the edges. But now these are opaque samples. And the motivating sample for this was when you start to think about nanofabricated surfaces in biology. Nobody wants to replace an SEM, and in a nanofabrication sense, why would you? But if you want a fast answer, or you want to put it underwater, or you want to see how it interacts with biology, that's a little harder to do with EM. And to be honest, the researchers that we talked to, it's a fair amount of work to do with confocal as well, because it does tend to like transparent samples. And so these were some nanoneedle patches from MCN, from Nico Wachler's group and Jan Harbitz, where they have, these are now not deposited, but etched surfaces of 200 nanometer cylinders. And this was the first little trial we did with them. Those are actually dried out cells. They're much more interested in doing this in water and doing it in a petri dish and looking at live cells. We also integrate with an off-the-shelf IVF heater, so you can do it at 37C. And so this is just an example where you might start to think, well, when you can't look at it because you want to do it in liquid, or you're starting to ask about sensors or that integration with biology, this gets interesting for opaque samples. Now, workflows in biology, because we are starting to scratch to that. And again, you can get great things of dried out cells in EM. Well, we can do this label free. So these are unstained samples. So this sample on the left are red muscle cells. They were fixed. And what you can see is there's no fluorescence here. That's just the HaloElement illumination. You can see the nucleus, the cytoskeleton, the little bright spots, forgive me, I didn't zoom in on this, are isosomes and little vesicles in the cells, where you can even see the cell membrane or the boundaries of that pretty easily. Now that's fixed. So I wanted to do something live. And the example I have is yeast. Now, no one on the planet needs another way to look at yeast. I know, great in bread, but everyone knows what yeast looks like. And so this is live yeast in solution, just moving around. And when I say that, it's because we actually watch these things move around in real time and watch the internals move around. And so you can see internals of live objects as well. And these weren't necessarily quite on the surface, but close enough to be illuminated with the evanescent wave. Now, if you ask, well, what are you trying to measure might affect your workflow? Fluorescence is used for chemical diagnostics, but sometimes you might want to know something other than that, or you want to know where things are sitting before you go through the work of staining and fixing or doing confocal. So this just did an idea of, you might start with bright field, and admittedly, if you have a fancier bright field microscope than the one we were using, you might get a little bit better resolution. But then that Halo image gives an awful lot of insight before you get to staining. And the confocal here again was done by the group from Melbourne with Sheeran, who's the PhD student, who just did a great job. And that's what she would do normally for her confocal staining compared to what the Halo image can deliver. And it depends on the information you're needing or where in your workflow you're looking. Is the sample viable? Is it going to look okay in confocal? Can I check that first? It also works in things like bacteria, much smaller. Normally you have to stain it. And this was a very high concentration. We're just trying it out. This was broth to slide in less than a minute. A drop of broth on a slide, slap a cover slip on it, and focus. And you can start to see E. coli and other cells. You can even start to see E. coli spit things off. And so again, this is an example where you can start to look at live material, live biological systems. And it really comes down to, what's the answer you're looking for? And that was a context that this group actually gave to us. Sometimes they might want to just know the shape of the bacteria. Is it breaking down? Is it spitting out small objects like outer membrane vesicles? Can you see that dynamically, particularly if you use this in a slide flow cell? But that group also, from their publications, does gorgeous confocal. That's the same wild type E. coli with a great confocal image. And cryo-TEM was a good example of trying to get that really fine detail on that cell membrane. So again, it starts to look like you could use these things for pre-screening or simpler answers, because sometimes you need a gorgeous, really high resolution detail. Sometimes you need less information or different information about a sample. And so that can sometimes fit in. So that was the biology part. And the last thing I want to do is just kind of finish up in the last few minutes with things that don't work, always work for surface sensitive systems, or things that just don't work for microscopes normally. So these are diffraction gratings inside a film. So it's inside a polymer film. It's actually a circulated $5 banknote. You'd never see this with a surface sensitive technique because this is in the film. Your best guess is in optical reflectometry, reflection microscopy. But these things are starting to get small, and these are the optical variable devices from the building on the $5 note. Now, you can see the color scheme there is a little different because some of the angles scatter very strongly to particular locations. Here is a case where you actually use the individual channel data, the light that's most perpendicular to whatever feature you're interested in, because it really gives a nice resolution of those diffraction gratings. You can pull them out, do measurements, Fourier transforms, get the pitch, and on the spacing, you're looking at like 475 nanometers for the spacing there. Now, we were able to do this on a circulated bill with nothing special. And that's actually a pretty powerful idea. So why does that work? So as I said, you can put particles on a glass slide. That's just an example of 200 nanometer particles in water, 195, sorry, on a glass slide scattering. But if you take a polymer film and put it on that waveguide, if you optically couple it, you'll actually generate the film at the top of that polymer film. But if you don't optically couple it, you'll actually generate the evanescent wave at that waveguide polymer film interface and scatter off what's there internally. And so that's how we were able to look at that banknote. Now, there's one more thing I wanted to show about the banknote. I noticed we have a quite international set of attendees, but for those of you from that are staying up late, Australian money is plastic money. It was developed at a combination of CSIRO and the University of Melbourne. It's got very interesting optical density character features, optical variable optical density features for anti-counterfeiting. It's used in like 30 countries. It's plastic. And your first impression might be it reminds you of Monopoly money because it's brilliant colors. But one of the things the Australian notes have are birds on most of them, and they look quite iridescent. And this is the bird, this little box of these flakes is the bird on the $5 note. Now, and it's either metal, clay, or plastic flakes embedded in that polymer matrix. The colors there, though, reflect the polymer, those flake orientations with respect to the particular locations on the halo. The flakes that appear black are the ones that are mostly flat. The other ones are all at angles. Now, this is still a 2D image, but it's encoding 3D information from tracking that angular information and that false color scheme we put in the lights on the software. So that's quite interesting about looking at a polymer film. And again, AFM, SEM would really struggle with these because this is not surface, this is internal structure. Now, the last one is, for those of you that have run microscopy facilities, you always get that special user that comes up with, hey, can I look at this under a microscope? And that was actually the discussion we had with Peter Sherrill at RMIT. He makes conductive or piezoelectric fibers, and that's his fiber mat. And he said, I'd love to look at that under a microscope. And it gives most of us shivers to think, okay, I guess maybe SEM on the top, we'll see. We're like, why are you interested? And he's like, it's a fiber sandwich. It's got thick fibers, thin fibers, and then thick fibers again. And he really wanted to be able to see inside of it. We said, okay, well, we'll give that a try. And so this is from the bottom of that thick fiber sandwich up until the middle, and that fiber sandwich. And at the bottom, we can start to see those thick fibers. It's not quite as dark field technique because you are getting a lot of scattering off all of that material, but you're still able to light pipe through the fibers and see them. And then as you get into the thinner fibers in the middle, as we actually here, we were slicing with a Z stack. We actually did it manually. It wasn't even a motorized stage at the best we could tell for equal increments. And what we're left with is we can see those really thin fibers in the middle, which are sandwiched between two thick fibers. So that would be reasonably inaccessible for a lot of microscopy methods without slicing it or fracturing it or something. And that alarm means I'm actually on time. And so I just wanted to finish to say, it was a hard choice on what to talk about. We've been working with a lot of different groups because there are a lot of applications for this platform. So if you do want to check out our website, which is just tinybrightthings.com, if you check out the applications part, there's 24 application notes of different ways we've looked at the HaloElement. And there's more you can read about how it works and details there, particularly in the resource page as well. But if you want to get in touch with us, you're interested in a trial or a demo or just want to chat more, please hit that get in touch button as well. So I'd like to stop talking and open it up for questions. And I
Oh,
might stop sharing.
okay. Well, thank you, Ray. That was amazing and absolutely fascinating. So let's, yeah, who has a question?